Good day to all the saints present. We can continue our studies on You're Fearfully and Wonderfully Made. We are fearfully and wonderfully made. He made them male and female, part 2b. Let's start by reading our text. Psalm 139, 14 to 16, and Genesis 1 to 27. I will praise thee, for I am fearfully and wonderfully made. Marvelous are thy works, and that my soul knoweth right well. My substance was not hid from thee when I was made in secret, and curiously wrought in the lowest parts of the earth. Thine eyes did see my substance yet being imperfect, and in thy book all my members were written, which in continuance were fashioned, when as yet there was none of them. And Genesis 1:27. So God created man in his own image. In the image of God created him. Male and female created he them. Fearfully and wonderfully made, he made them male and female. The object of this series is to show the genius of God in making mankind and to explain the text in Psalm 139:14, fearfully and wonderfully made, and the text in Genesis 1:27, he made them male and female. This is, in our view, very interesting information that believers can and should know in the context of our crazy, contrary, contemporary culture. In the first session of this series, we enjoyed a robust discussion after we tried to exegete Psalm 139, 14 to 16. In the first part of this segment, after discussing the aims and objectives of the entire series, we screened a compilation of videos that highlighted God's design in the marvelous miracle from conception to birth. In this segment of our program today, as we continue this discussion, let us explain the events during the first 3 weeks of human development or embryogenesis. And this will help us to appreciate the videos better when we view them again at the end of this session. First, fertilization usually occurs in the first third of the fallopian tubes. Fertilization can also occur outside the oviduct of fallopian tubes, but such events tend to end as ectopic pregnancies. When the spermatozoa binds to the zona pellucida of the oocyte, a protein called ZP3 acts as a receptor for the sperm in what is called the acrosome reaction. At the acrosome reaction that occurs when a sperm fertilizes an egg, there is release of acrosome contents. This is calcium-mediated enzymes that digest the zona pellucida, thereby exposing the sperm surface proteins to bind. As we can see in this picture, we have the corona radiata, the outside, and we have the zona pellucida. And the sperms are breaching into these two areas to get its head into the cytoplasm of the oocyte. And we see the similar thing over this side, and we see this sperm has been successful in breaching the cytoplasm of the oocyte. It is noteworthy that the head of the sperm carries mitochondria to generate his own energy to do that long swimming that he has to do. And it also has in the genes that is going to be transferred by the sperm. Oocyte membrane depolarization is caused by sperm membrane fusion, and this blocks polyspermy by preventing additional sperms from entering the egg. An enzyme alters the ZP3 protein of the oocyte so that it will no longer allow sperms to enter. So twinning is relatively rare, as you would then expect. Following fertilization, male and female nuclei approach each other and nuclear membranes break down. DNA replicates, the first mitotic division sperm contributes the centriole, which organizes the mitotic spindle, and there is zygote formation. Now you will see these things going on very, very clearly when we watch the video again, and you will appreciate them, what is going on, better than you did last time. The conceptus proceeds through a number of embryonic cell cycle rounds of mitosis, while still enclosed within the zona pellucida. The conceptus then progresses from a zygote to blastomeres, then to a morula, and finally to a blastocyst. The blastocyst, at about day 5, has two identifiable cell types, and a fluid-filled cavity called the blastocele, and an outer cell layer called the trophoblast. These peripheral flattened forms the placenta and placental membranes, and it also has the embryoblast, an inner cell mass, which is a mass of rounder cells located on one wall of the blastocele, which forms the entire embryo. Here's a pictorial summary of events associated with the first week of development. Our picture shows the orderly progression or the multiplication of the zygote, the fertilized egg. Here it is being wafted into the fallopian tube by the cilia in the fallopian tube. And as it proceeds along the fallopian tube towards the endometrium of the uterus, he is encouraged by the waft in the cilia of the fallopian tube. So the fallopian tube is designed to encourage the path of the sperm along its length, so it can be implanted in the endometrium of the uterus. Here is the same picture again, but with a little bit more detail. We see the zygote being formed. That is the fusion of the male and female contribution to this new life. And then here we see the 4-cell stage. And we see the morula about 3 days. It's called a morula because it looks like a mulberry. And that's the Latin for mulberry, is morula. So here we see the morula on its way to the lumen of the uterus. And by 4 days, it has got into the uterine cavity, is now called a blastocyst. And then it implants onto the endometrium of the uterus. The uterine tube epithelium consists of ciliated cells that are moving the secreted fluid towards the uterine body. The conceptus is floating like a boat within the fluid and moves in the same direction, as you will see and appreciate better when you watch the video. Towards the end of the first week, the blastocyst has reached the uterine body, and from about day 5 onwards, the blastocyst hatches from the surrounding zona pellucida. And we saw this very clearly in one of the videos last time. The conceptus can now receive nutrition directly and can commence the process of implantation in week 2. So, a summary again of what happens in that first week. We have cleavage. We have many cell divisions until 8-cell stage, to the morula, and then to the blastula, which, as you can see, the cells are on the outside, right? It's like a cyst. It is actually a cyst. A cyst is like a ball with fluid in it. So this is the blastocyst. This is what's going to be implanted in the endometrium of the uterus. So this is a lovely picture which says the same thing and tells us that we have come to the end of the first week of development, and we have a blastocyst, and within it we have the inner cell mass. This is the embryo, and we have the outer cell mass, which is the trophoblast. So here we have the trophoblast, and we have the inner cell mass. So we've come to the end of the first week. And again, we see the blastocyst approaching the endometrium, and we see it rolling down, and eventually it's going to reach out, invade the endometrium, and we find that we have forming what is called the bilaminar germinal disc. So this is coming to the beginning of the week 2, and we can see already that we have the hypoblast and the epiblast formed at the beginning of week 2. I hope that this additional information will impress you with the intricate design of the processes of the first week of the development of a human embryo. As wonderful as this is, think for a moment that God did not only design the process, but that he also supervises it daily all over the world. This is what David says in Psalm 139, 15 to 16. He says, my substance was not hid from thee, even when I was made in secret and curiously wrought in the lowest parts of the earth. Thine eyes did see my substance, even when I was yet being unperfect. And in thy book all my members were written, which in continuance were fashioned, when as yet there was none of them. David seems to be saying, or he was imagining, that God had a book with the information written down, and he sorts of tick off things as they are done. But that's amazing. That's an amazing thought. But we can't say that it doesn't happen. But I believe that he does supervise each birth, because that is what David seemed to have, information that David seemed to have got via the Spirit. So I'm not going to fight with you at all. Now we come to a very important point that we have to make about speaking to our daughters and nieces and so on. The danger of bad advice some girls give to their peers. There are many girls who will say to their peers at high school, I have been engaged in sex since I was about 16, and I don't use any contraception devices, and I've never got pregnant. Well, the truth of the matter is, such a girl, she cannot get pregnant because her tubes have been blocked because of her promiscuity. And we're going to go on to show the danger of this and why we must know these things and do serious teaching. In the last session, we learned that the vagina is protected from infection by the lactobacilli of Dulalat, that God has designed to live there naturally because these bacteria make acid. We learned also that God has designed the cervical also opening to be sealed off by a mucus plug. So you are now asking me, Doc, why then are there infections on the fallopian tubes, which we call salpingitis? Well, infection of the fallopian tubes are usually caused by ascending infection via sexual intercourse. We call it pelvic inflammatory disease. It may be caused by scarring from curettage with persons who have a previous abortion. The results of these things is ectopic pregnancy, infertility, tubal ovarian abscesses, and especially we can look at Fitz-Hugh Curtis violin string adhesions. Now, here we have for us on the left of screen a normal fallopian tube. And in the middle, in the lumen, we can see this is really these things that you see that look strange are really the cilia and the organization that is inside of the lumen of the fallopian tube to assist the sperm in its journey to meet the egg and in the journey of the zygote back down to the endometrium. But the picture on the right is a different story. We can see that the tube is enlarged because because of the inflammatory response, and we can see that the tube itself is blocked. It's really blocked with all of the exudates and the inflammatory response secretions that is inside the tube. This is not a good picture at all. And here is the real trouble. In such a case, the infection, if it is not treated drastically, you end up with these bands of tissue in the lumen of the fallopian tube. Now, these bands of tissue, this is very much like a grate. You have a billet, you have in the road, for instance, a grate. It will allow water to pass, small things to pass, but it will stop bigger things. So in acute salpingitis or in salpingitis or chronic salpingitis or as a complication of salpingitis, the young girl, her fallopian tube is like a grate, like a bar, like bars in a burglar bars. Now you can see that I love to talk about the blood supply to the ovaries, to the uterus. This is very important because this is going to come into vogue right now. On right of screen, you can see that the vaginal artery and the uterine artery, they both come off the internal iliac branch of the femoral, and we can see that they join up together. And if you hold your hands like this, that would be a good picture. We call it an anastomosis. They're joining up because they're gonna be going up sides of the uterus. They help feed the uterus with blood. And note that they're also anastomosed with the ovarian artery, which is coming off of the aorta. And here we see it again. In this, we saw some of these pictures, some of these strands on the uterus in some of the pictures we saw in the videos. And there's the same thing, the vaginal artery joining with the uterine artery and anastomosing with the ovarian artery. And here we see it even better. And we got a picture of the idea, the coiling that can occur alongside the body of the uterus in a pregnancy. Looks much like telephone wire, the telephone cord. So why is that so important? Well, you'll soon see. We all know that the fallopian tubes conduct over by ciliary movement of a ciliated columnar epithelium and periodic peristaltic contractions and fluid current from the surface of the ovaries to the uterus where fetal development occurs. But look now what happens if the fallopian tubes are blocked. Look what happens when our young female relatives or friends are given bad advice by their peers or engage in promiscuous living that ends in pelvic inflammatory disease and salpingitis and blocked fallopian tubes. Well, they end up with surgical emergencies due to ectopic pregnancy. And in this picture, we see the sperm, he swam and he has found the egg and he's coming back, but he is not allowed to get past that grate because of those Hugh Curtis adhesions. And just look what happens. All of this area we see, vaginal artery, uterine artery, ovarian artery, all this blood, this anastomosis, blood leaks from this. This is a lot of blood. This is a lot of trouble. This young girl could lose her life. Bad advice can lead to loss of life. And here is a picture of a fallopian tube which has been cut in half at post-mortem, and we can see two fetuses in the mass here. And again, here is another picture of an ectopic pregnancy with a fetus which has been discarded because they couldn't get past that. I will call it a grate, for better use of a word. But this is something that blocks its passage on its way to the uterine endometrium. And here we see another one. And again. So now we come to the second week of development. I hope that you've learned something from that last, those sections of slides, because this is important information that we must transmit to all people. In the second week of development, the formation of the bilaminar disc occurs. So just imagine two lines of cells. develop. That's all that happens in the bilamina in the second week, the formation of the bilamina disc and the completion of implantation. The second week of development is known as the week of twos because everything is divided in two in the second week. I believe this is divine order, organization, and design. I do not think that there is at all, this is at all happening by chance. This is very, very orderly thing that's happening right here in the second week. The embryo begins to implant in uterine lining at the end of the first week, and the second week of development is marked by and concerned with two things: blastocyst development and the differentiation of the blastocyst into early embryonic and placental forming structures, and completion of the implantation process. The cavity within the blastocyst and the inner cell mass that will eventually form the embryo begins early differentiation. The outer trophoblast layer grows rapidly and differentiates to form the embryonic tissue of the placenta. And we have a lot of pictures to illustrate this. So first, we see the, the, the trophoblast. This is the cells in yellow. And you see the inner cell mass within it. And it has approached the endometrium. And here, the syncytiotrophoblast and the cytotrophoblast has invaded the endometrium. Here we have the inner cell mass, which is going to give us the baby eventually. So this is the start of implantation. And here you see a similar thing. You see, we have the syncytotrophoblast, we have the cytotrophoblast, and we have the cell mass, the inner cell mass. This is what's going to become the baby, and it has something called the hypoblast and the epiblast. These two layers are going to become important parts of the body. This is very, very exciting. Implantation, which commences about day 6 to 7, is the migration of the blastocyst. Here's the blastocyst migrating into the uterine endometrial epithelium, and it's completed about day 9. So there's the interaction between the trophoblast cells and the endometrial epithelium. In this picture, we have cut the blastocyst in half, so you can see inside of it. It's a cyst, a big open space, and we have the inner cell mass, which is going to become the baby. Just this little thing here in blue is what's going to happen. It's going to become the baby, and here it is invading is outplanting to the uterine endometrium. And here comes the zygote. He's rolling down, becomes a trophoblast, has inner cell mass inside. Here he's be a little bit more developed after 2 days, and the syncytiotrophoblast and the cytotrophoblast is invading the lining of the endometrium, and we see this has happened a little further. And down here again, we see the formation of the epiblast and the hypoblast, and we see the formation of the yolk sac cavity. And I'm showing it to you a little bigger so you can see it a little bigger. Same thing, just a little bigger. And this is the result at the end of day 11. So we have two cavities. This is called the amnion, and this is the yolk sac. And we have here the hypoblast, and we have the epiblast. These two, this set of blue slight cells and this set of yellow cells are going to become two important parts of the body. Very exciting, I think. Again, in week 2, the two-layered embryo of the bilamar embryonic disc is formed when the inner cell mass divides into two sheets called the epiblast and the hypoblast. And these two make up the bilaminar embryonic disc. These are two of the things that happen in the second week, which we said is the week of twos. And again, The second week of development showing the embryonic disc. It's the embryonic disc, and it has the hypoblast and the epiblast, and it has the cytotrophoblast and the cytotrophoblast, and these are going to become the placenta and other things to support the conceptus. Again, I wish I had these pictures 10 years ago when I was teaching embryology. Here, since cytotrophoblast, the outside, the cytotrophoblast, within that, we have the yolk sac form, we have the amnion form, and we have the hypoblast, and we have the epiblast. Very exciting. Same thing. So if I could wake you up in the middle of the night and ask you what happens in the second week, you can tell me, Doc, there's a week of tools, and we have something called the cytotrophoblast, and we have the other blast, two sets cells, and we have this is the bilayer with the epiblast and the hypoblast and the yolk sac, right? And I said, okay, you can go back to sleep, pass the test, and here it is again, right? At the end of this class, you will know this without trying hard to memorize it, right? And there are two cavities that are formed in this process. And the reason why I'm going to show you this picture, because we will see these two cavities appearing over and over through the third week. Now, we tend to teach up to 4 weeks or 5 weeks in embryology classes, and then we go on to show how the respiratory system is developed, how the nervous system is developed, and so on and so forth, because by that time, by week 3, these things are well on the way. So when you go on and you teach it, you don't teach the general thing, you teach how each individual part is developed. So you will see these two cavities mentioned a lot, and in between is the embryo, just this little deceptive layer of cells. That is the bilateral layer that we are speaking about. That is so important in the second week of pregnancy. Now, if the mother is exposed to teratogens, anything, a teratogen is any substance that will be inimical to development of the fetus, she will lose the conceptus, right? So second week is very important. So by the end of the second week then, the blastocyst has undergone implantation and it has formed the bilaminar disc. This is very complex, but it is very orderly. This is the picture I had when I was teaching. That's all we had. Since then, a lot of good work has been done. But when my students saw this, they were very happy because they could see, your trophoblast is top coming from the blastocyst, and you got the hypoblast coming from the blastocyst as well. Sorry, from the inner cell mass. The blastocyst gives the inner cell mass, so you got the this and that. And it goes this and that, this and that, this and that, this and that. And they could see that. And that helped them to understand what they were reading in the books. But you can see this is very, 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 very orderly. This is very, very orderly. This is not something that is just happening so. And this is what happened about the end of the second week. We have this. We started here at the beginning of the first week, remember? And remember, we have the hypoblast, epiblast. We have our two cavities again. We have the hypoblast and the epiblast. We have our two cavities, and we have the supporting structures around, inside to your trophoblast and inside the trophoblast. And after that, we now come to form the body stalk coming from the yolk sac. Now, 5 or 6 weeks time, when we come back to look at the differentiation of the bipotential gonads, we will start with this piece here. So this is very important. So you've got very, very, We've done a lot today, and this is not baby talk. This is what they are teaching for the USMLE objective exams. Okay, so you are probably able to take some of the questions, answer some of the questions in embryology at the end of this class, right? I promise you. Here is where we are at the end of week 2. This is presented differently now because we have come to the stage where we want to talk about a special fellow. He's called the primitive streak, and he is going to take over and start to guide the next set of steps in the 3rd week. See him? The primitive streak. So we have converted this and show it like this. So we're just showing these two pieces, the blue and the yellow, right? That's the epiblast and the hypoblast. But the body, laid out like this in this part of the thing. And the primitive streak now is going to take over in direction. So we've entered the 3rd week. During the 3rd week of development, a 6-week period of rapid development of differentiation begins in which gastrulation occurs. Gastrulation is a process in which the bilaminar embryonic disc, formed in week 2, is transformed into a trilaminar embryonic disc by a process of rearrangement and migration of epiblast cells. So week 2, the week of 2s. Week 3, the week of 3s. Isn't that exciting? Gastrulation begins at the primitive streak, which is a raised groove on the dorsal surface or the upper part of the epiblast, and results in the formation of 3 germ layers. Note that all 3 of the germ layers are derived from the epiblast in the 3rd week. What happens now is that the inner cell mass, now called the embryonic disc, differentiates into a thick plate of ectoderm and an underlying layer of endoderm, right? The ectoderm is coming from the epiblast, and the endoderm is coming from the hypoblast. But the fellow is in control is the primitive streak. Primitive streak is taken over. From the primitive streak, cells spread out between what is going to be the ectoderm and the endoderm to form the mesoderm. This is the 3rd layer. The endoderm is formed from cells that replace the hypoblast. So we don't talk about hypoblast any longer. We now speak of endoderm. The hypoblast is converted to what's going to be the endoderm. That is the deepest layers of cells in our bodies. The mesoderm is formed between the epiblast and the endoderm. And the ectoderm is formed from epiblast cells that stay on the dorsal surface. And ectoderm is going to form those things which are on the outermost parts of our bodies, like our skin and nails and hands and so on. These 3 germ layers, which are the origins of many structures, begin to develop, leading to formation of the trilaminar germ base in the 3rd week of development. So here we see the primitive streak in action, and he is causing these cells from this area to spread out. And now we have 3 layers. We don't only have the blue layer, which we now call the ectoderm, and we no longer have the lower yellow layer, which we were calling the hypoblast before. He's now called the endoderm, and in between them, this red layer is the mesoderm. So we went from 2 layers to 3 layers. And here we see it. And here we see it in a different view. And again, right? We now have the endoderm, the mesoderm being formed. So we have 3 layers. And again, 3 layers. Beautiful pictures, these. And another thing turns up right here. Do you see him? This is the notochord. Notochord now, he is going to develop into the nervous system. He's going to supervise the development of the nervous system. And this happens by folding. Look at this green band here and this green band. Look at this blue band and look at this purple band. And notice that they fold. And as they continue to fold, eventually you have something called the neural tube. The neural tube is where we have the spinal cord. And on the sides, we have now the other nerves that go along the side of the spinal cord. So suppose we say that we start with a ball thread. We are pulling the thread out. And as we pull the thread out, all of these things are happening. Suppose the things on the thread were pictures. They were pictures, the film. That's what's happening. We're seeing these things happening. And some of these things we actually saw in the movies, in the videos, but they didn't explain it. So we try to explain it so you can be a little bit more comfortable. And here, the same thing. We started at middle of the week 3. We have the motor cord. See him here? And he's going to do his thing. And eventually, he is going to direct the formation of the neural tube. So what you see here, he's going to fold. And he's folding. The motor cord is in charge of this section. And this is what happens by the end of the 3rd week. We have the neural tube, and we have some spinal ganglions, what we call the autonomic nervous system, passes on the side of your vertebrae, and it sends branches to each somite, each branch of your body. Your body is divided somites, and each somite gets a branch coming off. Beautiful pictures, I must say. Again, there's the notochord, and there he forms a neural growth, and you will probably see this. Some of these things we actually saw, like this. We saw this in the videos, and you will see it again. And this is explaining the folding of the neural plate and the neural growth to form the neural tube. Right now, this is all very complex in a sense, but it is orderly, and that's why it is. We want to show you how complex it is, to show what a genius the Lord is, and the fact that this cannot happen by any big bangs. No amount of bangs will achieve this. This is too complex. But the fact that you can understand it and I can understand it, it is simple. In a way, you see, there's sort of all complex simplicity in this complexity in this design by our great God. So the 3rd week of development is characterized by the appearance of the primitive streak, the development of the notochord, which is going to go on to give us a nervous system, the differentiation of the 3 germ layers, which are going to give us these 3 germ layers are going to develop into all the different parts of our bodies. So in this 3rd week, what happens? We have gastrulation, that is the formation of the 3 layers. We have neurulation, which is the beginning of the formation of the nervous system. And up to now, the woman does not know that she is pregnant, even though organogenesis is well on the way. At 3 weeks, the heart is beating, the brain is forming, and some people are clamoring that we should get rid of this beautiful work that the Lord has done in this woman's body. Up to now, she doesn't know that she is pregnant. And all of these things have been going on. This is marvelous, marvelous things. To repeat, at the beginning, we had the zygote. Remember? The sperm and the egg met. We had cleavage, and cleavage, and we formed the blastula, which is really a cyst. It's more or less a cyst. And after, at the beginning of the 3rd week, this cyst underwent what we call gastrulation because of the influx or the invagination of cells and we form three layers, which we call the gastrula. Three layers. And those three layers are going to form all the three parts of our bodies. Again, so we have the blastostula is becoming a blastocele. It's a seal because it has a space in there. And then something called archenteron happens. It arches, right? And what we end up with the early gastrula. At the early gastrula, we only have the ectoderm and the endoderm, but by the end of gastrulation, we have all three germ layers. We have the mesoderm forming between the ectoderm and the endoderm. These three germ layers, which are origins of many structures as shown in table 1, begin to develop, leading to the formation of trilaminar germ. This in the third week of development. Here we are, and you can, of course, now recognize the notochord and the ectoderm. The outside part is going to be the outside part of the body, and the mesoderm is going to be the inside part, and we have the endoderm down at the bottom here, and the endoderm is going to form those things that line the innermost parts. And at this stage, you should be laughing because you don't have to learn this because you're not a medical student. All you have to do is remember that the gastrula has three layers. It has the endoderm, and the endoderm is going to form these things. You see, linings of the digestive tract, lining of the respiratory tract, pancreas and the liver, very, very, very intricate internal parts of the body, and the ectoderm, which is going to make outside things like the skin and hair and so on, and we have the mesoderm. So you can laugh. You don't have to learn this. You can just understand it basically. Isn't that fun? Back to the basics is lots of fun. And let me see it again. Remember, we had the epiblast, and we said everything comes from the epiblast. So we have the ectoderm, hair, nails, skin, and so on, brain and spinal cord, ectopart, and then we have the epiblast forming the mesoderm, and you see the things in the mesoderm, like the notochord, and the vertebral discs, the kidneys, and so on, and the endoderm, that's the epithelial linings and glands of the digestive and the respiratory tract and so on. And here he is again. Nice picture. The ectoderm shows you what is formed from the ectoderm section, what is formed from the mesoderm, and you can probably understand this better because it is shown, and the endoderm, the things that cover these structures and form these structures, heart and lungs and so on. Very exciting. So in week 1, we got ready for implantation. We made a blastocyst. In week 2, we had the bilaminar disc. In week 3, we had gastrulation. Primitive streak came and started the process of gastrulation. The neural plate was formed, and our nervous system was the formation of our nervous system started. Now we must state that, this is what's called organogenesis. In week 3, we start to make the organs. That's organogenesis. And this is susceptible to teratogens, especially in week 3 to week 8. And this is very significant because a lot of people, I have blogged with people who say, God made them so. Some people blame God wrongly for the congenital defects that sometimes occur. But these are quite rare when you consider the massive numbers of babies born all over the world on a daily basis. Many of these defects occur due to many teratogens. How many people you know in your village growing up that had defects? How many? See, how many of your friends or workmates that you know that the children have defects? It's rare. But people say, God did it. Teratogenesis is due to environmental exposures that can adversely affect the developing fetus. This is all the result of the fall. These adverse environmental exposures may include maternal conditions. The mother is taking alcohol and smoking and not getting proper nutrition in pregnancy. And she's got uncontrolled diabetes. None of our young girls should have rubella and they're pregnant because they should be getting the MMR vaccine, and certainly they shouldn't be getting syphilis, and they shouldn't be taking certain drugs, and so on. You see what I mean? These are things that can be avoided. These are terrorism that can be avoided. If mother's exposed to these things in the first 2 weeks, she will lose the conceptors. In weeks 3 to 8, you can have problems involving the heart, the upper limbs, the upper lip, and so on. Because we saw in the video, in the video, that these things are being made at this time. Now, after 8 weeks, it's not as bad, but you can have problems with your ears and your eyes and your teeth and so on. This is the same picture I just put here from a different book, different time. First 2 weeks, you're going to lose the conceptors. First week 3 to 8, heart is going to be affected, upper limbs are going to be affected, lower limbs. I think we showed in the videos the formation of the heart. That is a real beautiful thing, because you start with a sac, and there's a lot of folding that has to be done to get the ventricles and the auricles in the right shape, so that when the baby is born and takes over its own circulation, that the blood goes to lungs, come back to ventricles, and go push out and go to aorta and all that. That there's a lot of folding and a lot of complexity going on there. But that thing you could be in trouble if you have rubella, among other things. So a lot of things we can avoid. The big thing I want you to know about our first embryology class is that by the end of the 3rd week, the heart is working, and we saw it on the video. It is therefore not at all true, as recently suggested by a high-ranking judge, that a baby's heartbeat at 6 weeks is just a manufactured sound created for parents. That is not true, because at that time, we can hear the heartbeat. And you heard it on the videos, right? From Doppler to Doppler. This is the progression from week 11 to week 38. We saw this kind of thing on some of the videos that we watched. And here it is again. And I have to ask you 2 questions. Could all that we just described happen by any kind of Big Bang? Would you say that what we just described above indicates that we were fearfully and wonderfully made? Before we watch the videos prepared to bring the session to its zenith, let us give thanks.